there? Yes. We are live on YouTube, and so I think we can start.
Okay, good morning, everyone. It's a pleasure for me. I'm Elena, the Vice Coordinator of the Young Group of the Italian Chemical Society, to introduce you in this event. As you probably know, during the year we organize several awards, like the Levy Prize and the Reaxis Young Researcher Award. And we are used to celebrate our young researchers in big conferences, but this year, due to the COVID situation, we were unable to do that. But I hope that you can enjoy anyway this morning with us to celebrate our young researchers' success in these prizes. And it's a lucky coincidence because today there will be also the announcement of the Nobel Prize in Chemistry. So we will try to switch our live streaming event in Oslo to see who won this year the prize. But before starting this morning, which we will see, we will have the opportunity to listen to the presentation of each one of our awardees with 10 minutes presentation about their results, either in the PhD studies or in the postdoc activity. I would like to, first of all, thank our Elsevier partners. You can see Agne Carose, who is the Customer Marketing Account Manager for the South Europe, and all the other Elsevier partners that supported us even in this year to organize the Reaxis Young Researcher Prize. And I would like also to thank Professor Gaetano Guerra, who is with us. Gaetano Guerra is our president of the Italian Chemical Society, and I would like to give him the opportunity to say something to our young researcher, the brilliant young researchers that won the prizes.
Thank you, Elena. Can you hear me?
Yes, we can hear you.
I think for some reason you cannot see me, but I don't know why. I try to activate my video. Oh, now it works. Okay, now it works. Thank you very much to you for this invitation. Thank you to Marta Dapian and to you, Elena. On behalf of the Executive Committee, Comitato Esecutivo of Italian Chemical Society, let me say that we are really happy of these activities to support the activity of the young group. You have done many good things in the past and many good things we will make in the future. This is a difficult moment. We have to try to do the best to use these tools of online meetings. And these opportunities to combine also with the Nobel Prize winners announcement is very good, very, very good idea. So good work to everybody. I will be here to hear the presentation of young winners of these competitions. I think a good idea come from young people, and it will be very interesting for me to follow your presentations. Thank you again.
Thank you very much, Professor Guerra. So I think we can start the event. So before the award ceremony, in which we will present who won the Reaxis Young Researcher Prize and the Levy Prize, we decided to, let's say, to start this event by listening to a keynote with a speaker, Professor Monica Passananti, because she is a brilliant young Italian researcher who recently came back to Italy. So it's an example of the success that Italian chemists always do. Professor Monica Passananti is currently assistant professor at the University of Turin and also researcher at the University of Helsinki. So she works in these two institutions. She got her PhD title at the University of Naples on the photodegradation of pollutants in surface waters. And then she moved for a postdoc period before at the University of Blaise Pascal, and then in Lyon at the CNRS, and then at the University of Helsinki, where she spent quite a lot of her time, if I understood well. And she became interested in the study and modeling of cluster detection in the atmosphere. And as I said before, she recently came back also thanks to she recently won the ERC starting grant. So she started an own research activity about the impact of nanoplastic pollution in aqueous and atmospheric environment. So I hope she can present us not only the research activity, but also the pathway in which she got all this success and she won the ERC starting grant, because this is an example of, let's say, award. So it's perfectly on the topic of this morning event. Thank you very much, Monica, to be with us, and good luck to everyone, to all the speakers of this session. Thank you very much.
Thank you very much. Thanks for the introduction and thanks for inviting me. So let's see if you see. Okay, now I think you're in presentation mode. Okay. So what I will do is give you an overview basically of the environmental problem related to the nanoplastics. So actually, I will first give an introduction, and then I will try to answer some of the questions like where we can find nanoplastic, how many there are, and then I will talk about reactivity and finally some conclusion. So actually, I think you basically know that the production of plastics worldwide is really massive, and one of the problems is that it's increasing every year. So basically, for example, in 2017, almost 350 million tons of plastic has been produced. Just to give a visual idea on how much this amount is, actually, you can think that in 2015, 320 billion of liters, so in volume, of plastic has been produced. And this amount is equivalent to 125, roughly, the volume of the Great Pyramid of Giza. So you have to think this amount every year. And this, of course, leads to some environmental problems. One of the main concerns is actually related to plastics in the ocean, because most of the plastic finally ended up in the ocean. And also there, to give you an idea on how much plastics annually enter in the ocean, you can think that has been calculated that, for example, a truckload of plastics enters the ocean every minute. So at the end of this presentation, there will have been entered 15 truckload of plastics. Most of them, they come from the rivers, and most of the rivers that input plastic in the ocean actually are in Asia, but of course it's not only related to Asian country, but is a global problem. So actually, the size of the plastics that we can find in the environment can vary a lot from different order of magnitude. So we can have the big pieces of plastics, so the macroplastics, then smaller, until to arrive to nanoplastics, so where you have a size that is lower than micrometer. So in general, microplastic and nanoplastics are also classified according to their source. So we can have primary particles when they are directly emitted in the environment. For example, when you wash your face with scrubs, often inside the scrubs you have these plastic bits that then ended up in water and finally in the environment. Or actually they can be secondary particles. In that case, actually the plastics break, larger pieces of plastic, they break until to produce micro and nanoplastics. So let's move on where and how many nanoplastic we can have in the environment. So about where, well, I am sad to say that is really a global problem. So for microplastics, they have been found everywhere. It means in the deep ocean, surface of the ocean, atmosphere, soil, sediment, remote environment, mountains, snow, everywhere in the world, there are microplastics. And actually, for example, one of the reason is that they can move quite easily from one environment to another. And one of the main transport is actually the atmospheric transport. Indeed, microplastics have been found in a really remote environment where they should not be there. And actually they arrived there because of the atmospheric transfer. About nanoplastics, actually you can see from the plot that nanoplastic can also travel longer distances in the atmosphere because of course they are smaller, so they can stay much more time in the atmosphere and they can be transported over really long distances. This is quite a problem because then nanoplastic can spread everywhere basically, so they can reach any place in our Earth. So this is where the problem becomes global and not local anymore. So this is one of the problems related to nanoplastics. If we look at actually how many nanoplastics there are in the environment, also in this case, we do not really have a number because it doesn't exist yet a method that really is able to sample and analyze specifically nanoplastics. But actually, we know for the microplastics, and it's also quite logic, then when we decrease the size, we increase the number, because if we think just one bottle of plastics can be then fragmented in smaller and smaller pieces. So of course, when the pieces are smaller, the number increase. So the number can be huge. And one of the things that we can do, for example, is try to extrapolate the data for the nanoplastics. So in the plot, the red dots and the black dot are experimental data about microplastics in the atmosphere. So already from there, you can clearly see that the concentration, so the number of pieces, increase when the size decreases. So fitting this data and extrapolating the data for nanoplastics. So when you have the size that is lower than 1 micrometer, you can clearly see that the number of pieces increase exponentially. So there are really several order of magnitude higher compared to the larger pieces of plastics. So potentially the number of nanoplastics in the environment can be really huge. So after this introduction, I will talk a bit about also reactivity. That is another important aspect. And finally, about some conclusion. So about the reactivity, also in this case, the topic is quite new, so we do not know so much. But also in this case, there is one important thing to keep in mind. So for example, if we take a normal plastic bag and then we break this plastic bag in 40 nanometer particles, then we will expose a surface that is equal to 10 tennis fields. So actually, when we break a large piece of plastic in small pieces, we increase exponentially the surface area. And the surface area is where the reactions take place, because any kind of plastic from the bag to the nano react first at the surface. And then later, when there is some crack in the particle, then you can also have access to the internal part and core of the plastics. But the first reactivity is at the surface. So higher is the exposed surface, higher is the reactivity. So it's quite understandable that the plastic bag itself is almost not reactive, but if we have the nanoplastics instead of the plastic bags at this point, yes, we can observe a reactivity. So what kind of reaction we can have? So we can have a lot of reaction and interaction. Here in this slide, there is nothing about the soil, just focusing on atmosphere and water, so surface water. And if we made that all these orange dots are nanoplastics, actually they can interact, for example, with oxidant in gas phase, or they can interact with reactive species in liquid phase, and then they can also produce some degradation products. They can have a negative impact then on the environment. So now I will just focus on one tiny part of all these complex interactions. So it's actually the reactivity of nanoplastics with one of the main oxidants in the environment, so the hydroxy radical. So basically, the hydroxy radical is one of the major oxidants in the environment. He is produced by sunlight, and we can find him in the atmosphere, in surface water, and in cloud waters too. So what we have decided to do is actually measuring the kinetic constant between nanoplastics and OH radicals. We have used a competition kinetics method. So basically what we do is using a probe molecule, and we measure the reactivity of this probe molecule with hydroxy radical in absence and in presence of nanoplastic. When we have the nanoplastics, the degradation of this probe molecule is decreasing because actually nanoplastic can consume OH radicals, so they can scavenge it. So we did a series of experiments finally to calculate this kinetic constant for nanoplastics, in particular polystyrene, with different size. And actually this kinetic constant is quite important, because then we can use it for the in models to really understand if nanoplastics in the environment, they can react with OH radical, because OH radical is quite reactive, so it can actually react also with other species that are normally present in surface water or in cloud water. One example, for example, is the dissolved organic carbon. So what we did is comparing the dissolved organic carbon and OH reactivity with the reactivity between nanoplastic and OH. And actually what we observe is that, of course, depending on the concentration of nanoplastic and dissolved organic carbon, but in some case, actually we can have the reaction between nanoplastics and OH radical in the environment. So it means that actually nanoplastic can disrupt the normal equilibrium that you can find, for example, in surface water or in cloud water. Moreover, actually, we also observed that that this reaction, so the reaction between plastics and OH, can produce some products. They are released in the aqueous phase. And here you can see most of them are aromatic compounds, because the plastic that we studied in this case was polystyrene, so with an aromatic ring inside the structure. But this, of course, can be another environmental problem related to nanoplastics in the environment. So if we go back to the initial question, if nanoplastics is an invisible problem for the environment, I will say potentially yes. Potentially yes, because nanoplastic can be transported over long distances, so they can reach really also remote place. So the problem is global. Then another problem is related to the concentration, because actually the number of nanoplastics can be really high, because when a piece of plastics degrade is smaller than the number increase. And what increase is also the surface area, and so the reactivity. And actually, it's really this increased reactivity that can have some consequences. For example, we have shown that actually nanoplastic can scavenge OH in the environment, and actually this reaction can also lead to the release of organic compounds in aqueous phase. So potentially yes, but we need additional data. We need more people working on that, and there is still a lot of work to do. So one of the things that I really want to tell to all the young scientists is just, if you really have an idea and you think it's worth it to investigate that, go ahead. And it doesn't matter that no one is doing that, or it doesn't matter that it's something that you do not know what will lead after, but just try, follow your idea, and just do not give up, basically. So I want to thank all my collaborators, and also thank you for listening and for the invitation.
Okay, thank you so much, Monica. That was really an amazing presentation. It really opened up a lot of topics for people that are not acquainted with this topic. So really, thank you so much. And for the sake of the schedule, we will not have questions this time, as we won't have questions for any of the awardees, which I'm pleased to start with the announcement right now. So I will now share my screen in a second. And no, okay, it's not properly cooperating. Nope. It's fine, because we tried to share it with everyone before, and now with me it's not working. Okay, yeah, let's just go like this. Okay. Okay, you should be able to see my screen right now, right? Can you confirm that? I think so.
Yes.
Good, thank you. Let's proceed with what we are waiting for. So the announcement of the winners. And today we will have a combined award, as was said at the very beginning, because we will give the name and the prizes to two awardees, which are really, let's say, which we really have a feeling for within the committee, which are the Reaxis SCI Early Career Researcher Award 2020, so the edition of this year, and Premio Levi 2019. Both of them are historically being held and hosted by our group. And just to give you a few words about how the awards are meant to be, Reaxis SCI Early Career Research Awards started back 6 years ago. So we are at the 6th edition right now. And it's actually meant to award an essay that it's talking about an actual research project or a potential research project or a paper that has been already published on a research project, of course, and how Reaxis or other scientific databases have been useful to achieve that goals. We got 3 different prizes, and the first prize, it's so now I will proceed with the announcement. So the first prize, it's going to the presentation not working. Yeah. So first prize it's going to Giorgio Grillo from University of Turin, whose essay was entitled Scientific Databases and New Enabling Technology. So a big applause right now for Giorgio. And second prize right now has been awarded to Lorena Baranda-Pelliero from University of Rome Tor Vergata with an essay entitled Let Reaxis Be Your Research Ally. And the last prize, so the third position, has been awarded to Nicola Cavallini from Polytechnic University of Turin with an essay entitled Fantastic Online Databases and Why Use Them. So a big applause for all the three winners. And now let's proceed with the announcement of the Levy awardees. And again, just a few words about the Premio Levy, where Premio Levy is meant to award a research that has been conducted in Italy and that has to be original and of a broad interest for the chemical science and has to be published in an international paper before the beginning of this year, so in 2019. And now let's proceed with the announcement of this prize, which arrived at its 10th edition, because we started in 2010, the first edition. And so the first prize, it's been awarded to ex aequo, to a postdoc and to a PhD. And the postdoc who won the first prize is Simone Ranallo from University of Roma Tor Vergata, with a paper entitled Orthogonal Regulation of DNA Nanostructure, Self-Assembly and Disassembly Using Antibodies, which has been published in Nature Communication. Second prize, so still first prize, ex aequo, but for PhD category and second in the order of the announcement, has been given to Luca Capaldo from University of Pavia, with a paper entitled Visible Light Uranyl Photocatalysis, Direct CH to CC Bond Conversion, with a paper published in ACS Catalysis. Now let's proceed with the mention of honor that we decide to give to other two applicants. Again, one for a PhD category and one for the postdoc category. And the first mention of honor has been awarded to Stefano Corrà from Alma Mater Studiorum Bologna, with a paper entitled Chemical On-Off Switching of Mechanical Planar Chirality and Chiral Anion Recognition in a Two-Rotaxane Molecular Shuttle. And the paper has been published in the Journal of American Chemical Society. The second mention of honor for the PhD category has been awarded to Maria del Carmen Marin Perez from University of Siena. And her paper was entitled Enhancement of a Microbial Rhodopsin, the Electronic Reprogramming, again published in a Journal of American Chemical Society. And latest, we also decided to run a contest with a video that the applicants provided, and we decided to award the one that received the most not just click, but that was the most engaging with the public. So it was the most popular video. And the award for the most popular video has been given to Rosaria Bruno from University of Calabria with a paper entitled Multivariate Metal Organic Frameworks for the Simultaneous Capture of Organic and Inorganic Contaminants from Water, again published in a Journal of American Chemical Society. And now let's give a big applause to all the Levy winners and to the Reaxis winners. And to proceed with our award ceremony, now every winners will have the opportunity to explain to us and to show us in a 10 minutes presentation their research. Again, there will be no question afterwards. So let's start with the first winners to present, which is Giorgio Grillo from University of Turin with its essay entitled Scientific Databases and New Enabling Technology. So Giorgio, the stage is up yours. And I will stop sharing so you will have the possibility.
Hi, everybody. Yep. Thank you so much. Yeah, we have the presentation. Okay, before to start, I want to say a big thank you to the committee and to Elsevier for the outstanding prize. I'm very honored, and I'm very proud of the possibility to present my research today. I used the term enabling technology because to me this keywords means sustainability means a green way to think or to rethink chemistry. I work on vegetal biomasses valorization, and stealing some words from my PhD project, if we think about sustainable green biomass valorization, we should think about a cascade process approach. We should think about of every single component in order to reach a zero waste approach fitting inside biorefinery protocols. I tried to summarize our idea of biorefinery. If we think about starting from vegetal biomass wastes, we know that they are still rich in secondary metabolites, interesting because of their biological activity. We can recover them by means of extraction, but after that we still have a matrix composed by structural biopolymers such as cellulose, lignin, pectins, and so on. We can pretreat this matrix, we can convert this matrix down to the building blocks. And these building blocks can be furtherly modified by means of organic chemistry. Let's think about hydrogenation, oxidation, carbonylation, and etc. All these steps can be helped by the use of green solvents or catalysis, and of course of enabling technologies. I've reported here two of the main ones that I exploited during my PhD project, but today I would like to focus my attention on microwaves, and in particular between the four steps that you see here, I want to present you two case studies, both of them performed with the help of microwaves. Just a few words on the techniques. For the first case study, the extraction, we can say that the hypertonic solution inside cells is very responsive to the irradiation, so we can cleave the walls of our cells, releasing in that way metabolites inside our liquid, inside our solvent. About the second case study, we talk about platform modification, we talk about catalysis. If we think about an heterogeneous catalyst, we know that it is basically a support on which metals, nanoparticles are dispersed. In literature, it's possible to find this interesting article where the authors detected the hotspot right on the metal surface. So it is possible to enhance the energy transfer right where the reaction is supposed to happen. This is a very interesting point for process intensifications. But going straight towards the first case study, extraction of citrus peels. This residues is a very common waste in Italy. We worked with the CNR of Palermo. This is the work that we published after all the work. And this is a very quick overlook on the curve building on Reaxis. We got some outcomes, but with a very simple refining flowchart, it is possible to point our attention depending on what we are looking for. Industrial, if we need to check how these wastes are produced or exploited right now, or analysis if we need to design our lab approach. Merging all this information, it is possible to say that 50% of the weight of a fluid is considered right now a waste that actually is exploited as cattle feed. But also, we also know that these peels are rich in pectins and volatiles, and at the end of the extraction process, the residues can be furtherly applied for biopolymer conversion. We will see later something about it. The conventional production process, on the other hand, are not so clean. Let's just take a look to the acidic hydrolysis made by means of strong acidic conditions or the steam distillation requiring a high quantity of energy. That's why we try to apply our microwave systems, and in particular this microwave extractor, exploiting two setups, one hydrodistillation at the beginning and one gravity maceration at the end. After 1 hour, we got a two-step, one-pot extraction protocol able to double the pectin yield if we compare it to the conventional approach. This was an interesting starting point to us, but we are talking about wastes, we are talking about hugely produced wastes, so we need to start to face the scale-up of our extraction process. So we tested this industrial microwave extractor that basically merges the two setup, the hydrodistillation and the maceration. And at the end, we were not able just to reach our previous results, but we even enhanced it. We obtained 15% pectin yields compared to the 6% starting. But what about the waste of this extraction? What about the end of this the extraction? Basically, the residues are composed by cellulose, and this biopolymer it's very interesting, mainly because it is it's the main component of lignocellulosic biomasses, the most widespread vegetal biomass on the planet. So of course, finding a way to valorize every single component of this biomass should be a very groundbreaking result, of course. But today, we want to focus just on cellulose. It is well known in literature, hydrolysis processes that are able to obtain glucose that build the monomeric units by the application of enzymes or by the application of catalysts. So glucose then can be modified into a plethora of other molecules with application of an endless variety of organic reactions. That's why we decided to move to make a step forward and focus our attention on levulinic acids. This the procedure with which this acid is obtained from cellulose is quite well known in literature. And on the other hand, levulinic acid is one of the top 15 most valuable compound bioderived according to the US Department of Energy. In particular, we wanted to investigate the hydrogenation of levulinic acid towards gamma-beta-lactam, GBL, and pentanediol, PDO. This work was performed together with the University Ca' Foscari of Venice. This is the work that we published at the end, and going deeper in details, we investigated the application of gold catalysis with different hydrogen sources in neat conditions. Today, I will just talk about molecular hydrogen. And we studied two different supports, namely titania and zirconia. Once we find out that these two catalysts are comparable according to the metal nanoparticle dimensions and the metal surface activity, we tested them with the same identical conditions under microwave irradiation, of course. And surprisingly, we were able to see that titania, it's able to convert completely, more or less completely, our acid, giving some traces of PDO, something that zirconia is not able to do. We investigated by means of infrared analysis the phenomena, and applying pyridine as a probe, we understood that Lewis acidity of titania is higher compared to zirconia, and we hypothesized that this is the reason why we are able to activate the carbonyl group of GBL, and in that way you can go through the lacton ring and reaching the second step of hydrogenation towards PDO. we finally tuned a little bit our parameters, our reaction parameters, and finally we got complete conversion and selectivity towards our desired product. In need condition, we looked for this for similar reaction conditions in literature, and again, surprisingly, we saw that with gold catalyst and need conditions, not so many researchers were performed. And so I reported here the most comparable one, and even if authors here applied a more cheaper catalyst, you can appreciate the not so green solvent and even the substrate to catalyst ratio. It's not so sustainable. So up to now, to our knowledge, we can say that this is the first time in which levulinic acid was converted to 1,2,4 PDO over gold catalyst. So in conclusion, I can say that the possibility to exploit a so versatile tools like Reaccess, if you work with biomasses, it is fundamental, because if we think about vegetal wastes, we think about a huge quantity, huge variety of possibilities, and that works also for analytical approaches. So it is crucial to customize your analytical approach depending on the starting material, on the metabolites, and even on your lab possibilities. Just merging everything, if you work in green chemistry, or even more if you work with enabling technologies, it's crucial to have a solid literature benchmark comparison. In that way, you can say that you are changing the game, and in that way, you can pave the way to efficient and groundbreaking results. A very quick thank you to my mentor, Professor Giancarlo Cravotto, to Silvia, Manuela, and Maela, and to everybody that works with me in Turin. And of course, a big thank you to you all for your kind attention.
Grazie Giorgio anche per essere stato perfettamente in tempo, e adesso ci colleghiamo live con l'annuncio dei Nobel, che dovrebbe essere praticamente ora. Scusate, devo condividere con l'audio. Ok. Come vedete siamo in trepida attesa, dovrebbe andare appunto live adesso. Io direi di aspettare un altro minuto e poi eventualmente interrompiamo successivamente la talk dopo, in modo da mostrarlo, giusto per non perdere più tempo del necessario. A quanto pare sono in ritardo, quindi smettiamo questa condivisione e andiamo avanti con la prossima talk, chiedendo la cortesia che nel caso dovessero andare live mi permettete di interromperla per mostrare l'annuncio. Quindi passo la parola a Claudio.
Okay, good morning, everyone. I'm Claudio Imparato, and it's a pleasure for me to introduce the winner of the Primo Levi Prize. The first award, as already anticipated by Marta Dapian, goes to Dr. Simona Ranallo from the University of Roma Tor Vergata. So now Simona will tell us about her paper, Orthogonal Regulation of DNA Nanostructural Self-Assembly and Disassembly Using Antibodies, published on Nature Communications. So congratulations, Simona. And yes, we are ready to eventually interrupt if the Nobel Prize will be announced.
Interrompiamo già subito perché ci sono, quindi avevano poco ritardo, scusate.
Okay.
Allora, mando questo live.
Jointly to Emmanuelle Charpentier and Jennifer Doudna for the development of a method for genome editing. Die Königliche Schwedische Akademie der Wissenschaften hat heute beschlossen, den Nobelpreis für Chemie des Jahres 2020 gemeinsam an Emmanuelle Charpentier und Jennifer Doudna für die Entwicklung einer Methode für die Genomeditierung zu verleihen.
Perfetto, credo che siamo riusciti tutti quanti a vedere l'annuncio, forse non a sentire l'audio, però abbiamo visto i vincitori del Nobel e adesso ridò la parola a Simona in modo che scusate per l'interruzione.
Okay. Can you hear me?
Yes.
Okay. So thank you very much for the introduction, Claudio. Coming just after the Nobel Prize is I mean, just to go ahead. Anyway, first of all, I'd like to thank all the board of the young group of the Italian Chemical Society for this prestigious award and to organize this very huge competition for the 10th year. So first of all, I want to say that it's a pleasure to share this, sorry, it's not working. Yeah, to share this prize with my two co-authors. First of all, Daniela Sorrentino, she's a PhD student and friend, and she worked with me pretty hard. We're also having some fun working on this project. And of course, with Professor Francesco Ricci, that is mentoring me from a long time now. So I'm really excited to present here the paper that was published on Nature Communication Journal, and it's titled Orthogonal Regulation of DNA Nanostructure Self-Assembly and Disassembly Using Antibodies. So in this paper, we actually exploited antibody as molecular tool to guide the assembly and disassembly of nanostructure made of synthetic DNA. But why using synthetic DNA? This is actually a quite novel material that is finding a lot of application in the last decade, ranging from synthetic biology to DNA nanotechnology. In both these fields, our research group is focusing its attention, and we are actually developing many sensor, nanostructure, and nanoswitches activated by a wide range of inputs, but always employing DNA as a building block material. So many examples are reported today about DNA-based nanostructure that can be controlled and powered their assembly from different kind of inputs. For example, variation of pH. If we use pH-dependent substrates, so pH-dependent DNA sequences, or the assembly of this structure can be powered from small molecules or by proteins such as enzymes. However, it has not been yet demonstrated that antibodies can be used to activate and control the assembly of this kind of structures. And this would represent a very huge limitation in the field of DNA nanotechnology, but also in many other fields, such as clinical one, because it's well known that antibodies are important biomarkers for a wide range of diseases. So their molecular recognition plays an important role in many kind of applications, ranging from diagnostics to molecular imaging, antibody-based therapy, and targeted drug delivery. So our goal in this project was to try to fill this gap. Actually, in our research group, we are working and making a lot of effort developing antibody-based systems and circuits in the last years. Here, with this project, we actually started from the idea to exploit a well-known DNA-based reaction that is named toll-mediated strand displacement reaction. This reaction involves 3 DNA strands, 2 of them pre-hybridized to form a duplex complex over here that normally is optically labeled. So the activation of this kind of reaction that is visualized with the displacement and substitution of one of these 2 strands can occur just in presence of a 3rd strand. This 3rd strand is designed to displace and substitute the one that is labeled with a fluorophore usually. So actually the activation of this kind of reaction can be measured by fluorescence measurement with an increase of the signal. But okay, our goal was to use antibody. How to do it? We actually take this input strand that can be considered like the input unit and split it into DNA strands. Each of it was conjugated with a specific antigen. The idea is to design these 2 DNA strands able to have a complementary portion here in orange, but in a way that the binding affinity between these 2 guys is pretty low. Only when the antibody binds and recognizes the 2 specific antigen on the 2 DNA strand, these 2 strands will be confined in a very small volume, increasing the local concentration and allowing the reconstitution of the input unit. So only in this way we achieve in the activation of the strand displacement reaction, resulting in a release of a DNA strand. We have designed and characterized 2 DNA-based circuits activated and promoted respectively by the anti-dioxygenine antibody and the anti-dinitrophenol antibody. Both of the circuits show a very high affinity toward the specific target, and moreover, we are able, by varying the concentration of the input antibody, to modulate the activation and the response of this particular reaction. More interesting is also to see that these 2 DNA-based circuits can work orthogonally in the same solution without crosstalk. And this would be possible because we employ, of course, DNA-based system and circuits modified with different fluorophore. So the possibility to use these 2 DNA-based systems in the same solution lets us think, why don't use this kind of reaction to activate the formation of DNA-based nanostructure, but how to do it? Actually, the idea was exactly the one that I've just explained to you, but the DNA strand that will be released is designed to have a sequence able to activate small DNA monomers. These small DNA monomers are present in their inactive form. So only when the protector, so the strand release upon the presence of the specific antibody, goes to bind this monomer, they will be able to bind one each other, activating themselves and producing a DNA nanotubes. We have demonstrated even here that only upon the addition of the specific antibody we achieve in the assembly of the DNA nanostructure. And it's also interesting, as we have seen for these DNA-based circuits just working in solution, that we achieve an antibody concentration dependent behavior in terms of the length and the number of the DNA nanotubes formed in presence of the specific antibody. Even in this case, we have adapted this kind of system to a second antibody, the anti-dinitrophenol antibody, achieving a comparable result. So this kind of nanostructure that can be of utility, this is a proof of principle, but they can be of utility for a wide range of applications, including the drug release one. It's really important to see how they can work orthogonally in the same solution. So we can achieve, respectively, the activation of just one of the 2 DNA nanostructures of both of them just when the antibody are present at the same time. So the possibility of employing these two DNA-based circuits in the same solution let us think to try to make and design new DNA-based circuits able to activate the assembly of the specific DNA nanotubes in presence of a particular antibody, and then also to modify this design in a way that when the second DNA-based circuit is present and activated by a different antibody, we can actually release a DNA strand able to disassemble the same structure. So in conclusion, with this paper, we have demonstrated that we can control the assembly and disassembly of DNA-based nanostructure using specific antibody in an orthogonal way. This approach is in principle really versatile because can be generalizable to any antigen that can be attached to a DNA anchoring strand. And just as with Lego bricks, we can actually play with DNA to build DNA-based system, DNA-based nanoswitches that can find application in a wide range of field, including drug release, point-of-care diagnostic, and in vivo imaging. So last but not at least, I'd really like to thank again all the board of the SCI Giovani to honor me of this prestigious prize, and all the professor, Daniela, all the group of analytical chemistry of the laboratory of University of Tor Vergata, and of course, all of you for your kind attention.
Thank you very much, Simona Ranaldo, and again, congratulations. And also I want to congratulate with all the winners. And now I'm glad to introduce the winner of PhD category of Premio Primo Levi, the Dr. Luca Capaldo. Okay. And he will present us his awarded work with the paper entitled Visible Light Uranyl Photocatalysis, Direct CH to CC Bond Conversion, published on ACS Catalysis.
Perfect. Can you hear me?
Yes.
Perfect. So first of all, let me thank the scientific committee for giving me this incredible opportunity to show my data here in the ceremony, and of course for awarding me this prestigious prize that will be, I know, very important for my academic career. So my name is Luca, and today I'm going to be talking about the uranyl cation as a visible light photocatalyst for CC bond formation through hydrogen atom transfer, HAT. So when I started my PhD, I basically fell in love with photocatalysis. And this is a branch of chemistry that studies how a photocatalyst interacts with a substrate. But what is a photocatalyst? It is a species that you purposely add into the reaction mixture in order to convert light energy into chemical energy for substrate activation. And this activation can occur according to three different mechanisms. The first one is single electron transfer, SET, where the excited state of the photocatalyst just oxidizes or reduces the substrate to get the corresponding radical ions. And then these are responsible for the observed reactivity. Then we have hydrogen atom transfer, HAT, where the excited state of the photocatalyst cleaves homolytically a CH bond embedded into the substrate to get neutral cis-center radicals to be adopted to explore molecular complexity. And then energy transfer, ET, where the excited state just donates energy to the substrate to get the corresponding triplet excited state to be used in synthesis. But what struck me the most when I started looking up literature at the beginning of my PhD was that while SET and ET are routinely run under visible light irradiation, HAT was fairly limited to UV light irradiation. Visible light irradiation is cheaper and more sustainable. So actually, some records started appearing in the literature reporting visible light photocatalyzed HAT. For example, an alylation reaction was reported by Murafuji in 2016, where he used this complicated conjugated aromatic ketone called pentasine tetron to activate cyclohexane to get the cyclohexyl radical that he trapped with this olefin here in a radical addition reaction, 70% yield. Then an alkylation reaction was reported 2 years later by Wu, where he used using Y to activate cyclohexane and achieve its alkylation, even if it was just a proof of concept work instead of a preparative one, as you can see from the low yield. And then we have a fluorination reaction reported by Sorensen in 2016 again, where he used this uranyl dication nitrate to activate cyclohexane and achieve its fluorination in the presence of a fluorinating agent, NFSI. So based on our previous knowledge and expertise on oxo complexes to run HAT transformations, I started wondering if we could use the uranyl dication to form CC bonds under visible light irradiation. So I went to the lab, I took some uranyl dication, and I recorded some absorption and emission spectra to check. I mean, the literature tells us, but I wanted to check if the photocatalyst was visible light absorbing. Then I picked a model reaction, which is the same reaction that Wu reported in 2018. And I started screening all the conditions. For example, I found that 456 nanometers was the optimal wavelength of radiation. Then PC loading had to increase up to 8% to achieve full conversion of the limiting reagent, which is this olefin, styrene-like trap olefin reported here. Then the solvent was acetone, was the best one. I had to increase reaction time up to 24 hours. And since the excited state of my photocatalyst was not being quenched by molecular oxygen, I was allowed to run my reactions in air-equilibrated conditions. And then, of course, blanks were blanks, so light and photocatalysts were mandatory for the process. Here is the optimized conditions. So as you can see, the yield is quite high, 96%, almost quantitative. And these are the experimental setup that I use in my project. So on the left, you can see the explorative experimental setup, where I use this case equipped with a Kessel lamp, 156 nanometers, as I said, 40 watts. And you have this tray with 8 spots, so you can run up to 8 experiments in a single run to explore the chemical space of your transformation. And then the preparative experiment is reported on the right. You can see that we use these duct-like glass vessels below a Kessel lamp, just 5 centimeters away. And this is to isolate the products that are reported in this slide. Now, I cannot go, of course, into detail of each single product here, but I just want to focus your attention on some particular and representative entries. For example, I was able to functionalize cycloalkanes, alkanes, ethers, acetals, alcohols, amides, aldehydes, and so on. But the most interesting part in this slide is the right part, so the scope of the olefin. As you can see here, from entry 18 to 20, the substitution pattern on the aromatic ring of the olefin is not that important for the reaction. As you can see, the yield doesn't change that much. But if we change the electronic nature of the substituent in the para position of the aromatic ring, you can see that reaction times get longer and longer. And the same effect was reported for the substitution on the alpha position of the styrene-like olefins. Then in the last part of the work, I focused on non-aromatic traps such as conjugated nitriles, esters, and ketones. Of course, the second part was a mechanistic part, so a mechanism investigation. We still need to prove that we have an HAT process. So I ran some deuteration experiments. Basically, you have to run your reaction under your optimized conditions. Instead of using cyclohexane, which is the H donor, use an equimolar mixture of protiated cyclohexane and deuterated cyclohexane. What you have to calculate is the ratio between the product containing the protiated cyclohexane and deuterated cyclohexane after irradiation. And the ratio gives you the kinetic isotope effect, which is that value that tells you if it is an HAT process. If this value is higher than 1, it means that a hydrogen transfer is being the rate determining step in the process. So in accordance with an HAT process. I ran a parallel experiment, completely different experiment, photophysical experiment, that told me basically the same. So if you monitor the emission profile of your photocatalyst, upon addition of increasing amounts of the quencher of the excited state, on the left you can see here the decay in the presence of increasing amounts of proceated cyclohexane, on the right deuterated cyclohexane. You can see this decay. You can follow the decay at 508 nanometers, for example, and then if you plot this decay against the concentration, you get the kinetic constant, the ratio between the kinetic constant obtained with proceated cyclohexane divided by the deuterated, the one obtained with deuterated cyclohexane as the quencher, you get the kinetic isotope effect. As you can see here, these values are very similar. And these, both of them, tell us that this is a hydrogen atom transfer being run under visible light radiation. So here is the mechanism we propose in the paper. So you start with the uranyl dication. Of course, you absorb a visible light photon to get the excited state. Then you cleave a CH bond embedded into the substrate to get straight neutral radical, which is then trapped by the electron poor olefin to get this radical duct that is accountable for the closure of the photocatalytic cycle. So I have not enough time to tell you that these parts of the closure of the photocatalytic cycle took kind of weeks to be completely understood. But anyway, to conclude this mechanistic picture, we had no hints of radical chain mechanisms. So in conclusion, I hope to have convinced you in this super fast presentation that uranyl dication is a robust and efficient photocatalyst for visible light HET, and in general, photocatalysis an extremely powerful tool for substrate activation in organic synthesis. The uranyl dication helped me to activate really tough CH bonds, such as those embedded into cyclohexanes, which are routinely used as solvents in the lab because they're considered completely inert. And the most important feature of this project, this protocol, is that it is operationally simple. You just mix everything up in the reaction vessel and then shine light on it. This is something that every synthesis practitioner or organic chemist wants. The reaction needs to be operationally simple. To close, I would like to thank my research group, the Photogreen Lab, especially my mentor, Maurizio Fagnoni, and my co-workers in this project, Davide Ravelli and Daniele Merli, and of course, all of you for your kind attention. Hello?
Okay. No. It's okay, Marta?
Yes, yes.
Okay, sorry. Okay, thank you for your presentation. Now is the turn of another awarded, that is Lorena Barando Pelejero from the University of Tor Vergata, Rome, in the group of Professor Ricci. She got the second prize for the Reaxys Early Career Award, and the title is Let Reaxys Be Your Ally. So, please, Lorena.
Okay, yeah, I'm sharing the screen. Okay. So good morning, everybody. My name is Lorena Baranda, and I'm a second-year PhD student at the University of Rome Tor Vergata. And today in this presentation, I'm going to talk about the field in which I'm doing my PhD work, which is structural and functional DNA nanotechnology, and how I can apply Reaxys into that. So the field of DNA nanotechnology takes the molecule of DNA out of its biological context. So DNA is not used because of its abilities to carry the genetic information, but because of its chemical and physical properties, such as the well-known base pair interactions, the ability to self-assemble, and this together with the low cost of synthetic DNA, it's possible that we use this molecule as a tool for nanotechnology. So there are mainly two branches inside of this field: structural DNA nanotechnology, in which DNA is used as a construction material for the generation of DNA nanostructures, and also functional DNA nanotechnology, where DNA can be used for sensors or biomarkers, or also to control chemical reactivity in solution. So in this presentation, I wanted to give an example of how we can use Reaxys for a project involving DNA nanostructures, and also a project that I recently finished on DNA template synthesis. So let's go first for the DNA nanostructure one. So thanks to the biocompatibility of DNA, DNA nanostructures can be used for smart drug release. So these structures can be used as vehicles to transport cargo to the site of interest. We can have this closed state and this open state where the drug can be released. Several examples have been already reported on that, and we can see that we have chemical modifications in these DNA structures outside, so as they can sense the environment, and also they encapsulate a drug inside. So if we are interested in working on a project similar like that, we need to select these modifications. So for instance, we can focus on the selection of the drug, and here is where Reaxys comes to give us a hand. So Reaxys has this huge database that we can surf into thanks to the search engine. So I started by doing a broad search by selecting the target that I want to use, that I want to treat with my drug. So in this case, I selected SARS because of the hot topic of this year, so respiratory syndrome viruses. And then I could further refine this search by selecting the type of protein within the virus that I'm interested in targeting, and also the action on this target, in this case, inhibition. So then Reaxys gave me this panel where it shows the targets that meet the requirements that are previously specified, and also the drug candidates against each of these targets. Then I could further refine this search by only selecting the marketed substances, so as I could skip all the drugs that are still under clinical trials. And also, when I selected the drug that I'm interested in, I could go into the main properties of the molecule and also the main results that were obtained regarding these molecules, so the main in vitro and in vivo studies. So with all this, I wanted to show how we can use Reaxys for the selection of the modifications or the drug that we want to encapsulate in the DNA nanostructure. Now I'm gonna quickly get into how I applied also Reaxys to a project that I recently finished. So this project was involving the field of DNA template synthesis, where DNA is used to control reactivity. So the reactants are linked to DNA sequences, so that only the sequences that are designed to be complementary will do so and then produce the product of the reaction. For my project, we wanted to go one step further and control these reactions by antibodies. In order to do that, we introduced antigen modifications into the DNA strands, so as only in the presence of the antibody, we will have the hybridization of the strands and then the reaction and the product formation. This work was recently submitted with the name Using Antibodies to Control DNA Templated Reactions. And in order to select the reactions that I could template with these antibodies, I went to Reaxys and then I typed one of the most famous ligations in between DNA oligonucleotides, which is phosphoramidate DNA ligation. And then I came across this paper as a top result that showed me the modifications that I need in the DNA, also the substances that I need, the reagents that I need in solution, and all the conditions. And also it was talking about another reaction, which is the copper catalyzed click reaction. So I also went and searched about this reaction, and I got quickly the conditions and again, the substances in order to do that. Once I got all this information, I went into practice, and I started by the copper catalyzed reaction. Then I introduced a side and alkyne into my DNA strands, and then ran this reaction in the presence and in the absence of this antibody, hoping that I will only get this product in the presence of the antibody. The way to characterize these reactions was by denaturing electrophoresis, because the product runs as a band way less because it has double the length compared to the starting oligos. And then I saw that in the absence of the antibody, I do not get any product, whereas in the presence, I get this product here. So the strategy worked. Then I switched to the phosphoramidate ligation. Again, following the conditions that I found, I changed the chemical modifications for amine and phosphate, and I ran the reaction again in the absence and in the presence of this antibody, and I got a similar result. So with this, I wanted to show as well how reactions proved to be useful for my research in this project. And with this, I wanted to thank all the members of my lab, and especially my supervisor, Francesco Ricci, also the ITN DNA robotics in which I am involved, and of course, Società Chimica Italiana and Elsevier for organizing this award and then
Thank you, Lorena, for this interesting research. Now we switch to Premio Primo Levi, and I am happy to introduce the honorable mention for the postdoc category to Dr. Stefano Corrà from University of Bologna, with his work entitled Chemical On-Off Switching of Mechanically Planar Chirality and Chiral Anion Recognition in a 2-rotaxane Molecular Shuttle. This paper was published in Journal of the American Chemical Society. Please.
So thank you, Chair, for the kind introduction. And first of all, let me thank the organizer of this symposium for giving me the opportunity of presenting our work in this context. So the story I'm going to tell you deals with two concepts. One is the chirality, with which we are all more or less familiar, and the other one is the mechanically interlocked molecules, so entities in which two distinct molecular components are interlocked together. And a question, since the mechanical bond limits the possible orientation of the two components in space, a question arose already 30 years ago. So can we make molecular entities in which chirality arises simply from the relative orientation of two separated interlocked components? And the answer is yes, and in particular, in the case of rotaxane, if we consider of taking an axle in which there are two different extremities and thread it through a ring in which the only element of symmetry is a reflection plane which contains the ring, and this is so-called an oriented ring, you will soon realize that there are two ways that this threading can work. And the two molecules, the two mechanically interlocked architecture that you obtain are mirror images non-superimposable. So they are enantiomers, even though the starting components do not display any covalent stereogenic elements. If we now imagine of taking this concept even further and imagine of interlocking, of threading a symmetrical axle into an oriented ring, so an axle in which the two extremities are identical, as long as the ring is sitting in the middle of the axle, this structure is non-chiral. However, if we imagine of moving the ring towards either end of the left-hand side or the right-hand side of this symmetrical axle, the position of the ring desymmetrizes the molecule upon co-conformational switching, and therefore these two co-conformations that we obtain are enantiomers. They are, you can see, they are mirror images. This has been for the first time demonstrated in 2017 by the group of Saito, in which they were able to block an oriented ring on one side of a symmetrical axial by introducing a blocking group in the middle of the axial. And what they demonstrated is that what you obtain is in fact an enantiomeric mixture, a racemic mixture of two mechanically planar rotaxane. However, this system is non-dynamic, so there is no simple way with which the oriented ring can, one enantiomer can interconvert in the other, because we introduced a blocking group in the middle. So our aim was to create a proper stereodynamic system in which the two enantiomers can interconvert with each other upon a certain stimulus. And we envision that a bistable rotaxane with two identical stations that could accommodate a dibenzo 24 crown 8 ring would be a good scaffold for performing this task. So if you imagine of taking a ring in which a symmetrical ring in which this R group is a proton, you will see that upon threaded to an axial with two identical stations, you will realize that shuttling of the ring between the two stations converts one co-conformation into another, but these two co-conformations are identical. And this allows us to study the process of shuttling of the ring. If you now consider to do the same thing, but with an oriented ring, so with this R group that is not a proton anymore, what happens is that interconversion between the two co-conformations is not a simple shuttling process, but is converting one enantiomer into another. So effectively is a racemization equilibrium. So what we did practically was to synthesize two rotaxanes, prochiral rotaxane, in which one with the symmetrical ring and the other one with the oriented ring. Upon deprotonation of the ammonium station of this rotaxane, we force the ring to move towards one of the two secondary triazolium stations. And so we obtain the bistable rotaxane that we designed. The ring is constantly shuttling between the two stations. So what we did in the beginning was to compare the activation parameters between the symmetrical and the non-symmetrical version of our rotaxane, and we soon realized that the architecture of the ring is not affecting the shuttling equilibrium. If we now focus on these methylene protons that are marked in red in the picture, those protons are enantiotopic as long as the system is prochiral, namely the ring is sitting at the center of the axial. They are enantiotopic, so in the NMR, they resonate at the same frequency. They are completely identical. However, when we desymmetrize the system, so we deprotonate our rotaxane, these two protons are not enantiotopic anymore. They become different. They are diastereotopic, so-called. And this means that in an NMR spectrometer, they should resonate at different frequency and being coupled with each other. And what we did was to demonstrate exactly this. Upon slowing down the ring shuttling equilibrium in the NMR time scale, so going down with the temperature, we saw that the signal marked in red splits up from a singlet at room temperature into an AB system. So two different signals coupled with each other, which demonstrates that this molecule displays mechanically planar chirality in the deprotonated form. Moreover, we also confirmed that the racemization process, so the conversion of an enantiomer into the other, proceeds via ring shuttling. So I have just told you that this rotaxane with the oriented ring exists in solution as a racemic mixture of two enantiomers that are constantly interconverting between each other. If you now consider that this molecule is a dication, in the solvation sphere, at least of the non-encircled station, there must be an anion. All the experiments that were performed before were performed on the molecule that as it comes from the synthesis, in which the anion is an iodide, so achiral. And in this case, there is no way of inducing a differential population, so an enantiomeric excess, into the system. However, what happens if we imagine of introducing a chiral anion? If we put a chiral anion in the system, the ion pairs that we obtain in solution are diasteromeric, and so they are different. They have different stabilities, and so in principle, we can imagine of make one of these two diastereoisomers more populated than the other, or in other words, we can imagine of differentiating the stability of the two identical stations, and by this induce an enantiomeric excess in the rotaxane. So this is exactly what we did. Let's now focus our attention on the proton, which is marked in purple. As you can see, it appears as a sharp singlet when the anion is a chiral, the iodide. However, when we introduced optically active, strongly coordinating anion, which is camphor sulfonate, we could see that this resonance and some other resonances as well splits up into two sharp singlets, very close but distinct, with a ratio of 85 to 15. And this confirms that the two different diasteromeric ion pairs are formed, and the presence of a chiral anion bias the shuttling equilibrium of the rotaxane towards one of the two mechanically planar enantiomers. So, in conclusion, I hope I could show you that we were able to prepare a proper stereodynamic system where chirality comes solely from the relative orientation of two non-chiral components, so from the presence of a mechanical bond, and this exists as two enantiomers in solution which interconvert in each other via ring shuttling. This is the first example also of reversibly switchable chirality in a rotaxane. We can turn on and off the chiral character of this architecture by a pH stimulus. But more importantly, this is the first example of a dynamic stereoinduction in a rotaxane induced by introduction of a chiral information, so by chiral anion binding. And this brings me to the acknowledgements. First of all, I would like to thank the entire Credi group, but in particular Professor Alberto Credi and Dr. Massimo Baroncini, as well as Dr. Christian Devet, which started this research and with whom we had plenty of discussions. The University of Bologna, the CNR, and the European Research Council for funding this research. The organizing committee and the scientific committee of the Scientific Chemical Society for giving me this honorable mention, which is not as prestigious as the prize, but anyway, it's a great honor for me to have received it. Thank you very much. And I would like also to thank you all for your kind attention.
So thank you, Stefano, for your presentation. And now let's move to the other category, the PhD category, of the honorable mention of Premio Levi 2019 with Maria del Carmen Marin Perez from University of Siena, and she was awarded on the paper titled Enhancement of Microbial Rhodopsin via Electronic Reprogramming. Please, Maria, the floor is yours.
Okay, thank you. Good morning. First question, can you see my screen?
Yeah, we can see it, but please exactly just put in the presentation. That's perfect. Thank you.
Okay, let's start. Thank you so much. Well, first of all, thank you to the organizer to give me this opportunity to present my research here. This was supposed for me a great opportunity and also one more step in my career as research. So the aim of the research that I am presenting today is one of the most recent applications associated to the automatic rhodopsin modeling protocol for the automatic generation of computational models to study spectroscopy and photochemistry reactivity of rhodopsin proteins, and novel technology called optogenetic. This protocol was developed in the Professor Massimo Libucci group in 2016, and now there are new versions that are available for everyone. So let's talk about optogenetic. Optogenetic is defined as a biological technique which involves the use of light to control cells in living tissues, typically neurons, which has been genetically modified. In this technique, the scientists are interested to excite, inhibit, or visualize the action potential using light. Very briefly, this action potential is the temporary change from the negative to positive of the neuron membrane voltage. In nature, this potential is generated by chemical signal from another axon. So the aim of the optogenetic research is the development of molecular tools to excite this action potential using light rather than chemical signal. Of course, they are interested to have the full control of this technique, developing tools that also can inhibit this action potential. However, and this is going to be the main subject of my presentation, what is really important nowadays is the development of tools that can visualize the action potential. And this can be done using fluorescence proof. Research is related with rhodopsin proteins because they absorb the light, generating the photoisomerization of the double bond of the prosthetic group, the protonated shift base of the retinal chromosome. This isomerization can be used to activate, inhibit, or visualize the action potential that previously we described. And how do we do that? Well, the chromophore, which is inside the protein, is irradiated with light. It goes from the ground state to the first excited state, and then exploring the potential energy surface, come back to the photoproduct, passing through the conical intersection. So in principle, if one want to maximize this fluorescence, the isomerization of the retinal chromophore should be slowed down or totally blocked. For example, creating a potential energy barrier in the first excited state, as I am showing you in color red in the slide. At this point, and as we described previously, photoreceptor proteins are frequently used as biotechnology tools in the genetics to control the activity of optical cells. One of the main protein family for those tools are microbial rhodopsin. A good candidate to screen this fluorescent proof, and as I will explain you in the following slide, is anavenous sensory rhodopsin. From now, we will call it ASR. Now the question is, which are the motivation to employ this anavenous sensory rhodopsin in this screening? Well, the first motivation is we have an X-ray crystallographic structure available. So for us as computational chemists, it's very important to have this X-ray crystallographic available. In the case that we don't have it, we can generate a comparative model from another protein with a very similar or with a high percentage of amino acid sequences. The second motivation, and I think this is the most important for me, is that during our bibliography research, we developed that this anavenous sensory rhodopsin presented a dim fluorescence. So it is a very good starting point for us to study this protein because we can generate new mutations that can improve the current fluorescence observed. The last motivation for us was that then our external collaborators, we have available experimental data, not only absorption maxima, but also, for example, excited state lifetime, fluorescent measure. So it is very useful for us because we can compare our result and check if everything is going in the correct way. We study and among all the mutants that we generated of an adenosine rhodopsin, I would like to catch your attention on two specific mutants. One is single on the left part of the screen, L83Q, and the other one is a double mutation on the right part of the screen. It is double B76SY179F. Generated with our automatic rhodopsin protocol using the wild-type model as initial structure. The selection of these two mutations was not something casual because in previous experimental paper, we observed that these three positions in the amino acid cavity around the retinal chromophore presented an increase of the absorption maxima, and it helped us to start to think that maybe it can help in the increase of the fluorescence. Both mutants, as you can see in the table and also in the figures that I can show you right now, presented computationally and also experimentally blue shift effect of the absorption maxima with respect to the wild-type model. However, and as I shall show you in the figure on the center of the picture, these mutants has an opposite behavior with respect to fluorescence. While the single mutant presents a negable fluorescence with respect to the wild type, the double mutation presented one order of magnitude increased with respect to the wild type model. So what is happening at the molecular level? Let's discover it. To understand how the mutants can control this fluorescence, for example, creating an energy potential barrier, we calculated the energy profile of all these mutants along the isomerization coordinate. We found that, as we thought, the most fluorescent mutant, the double one, has a higher barrier in the first excited state than the wild type, while the less fluorescent mutant, the single one, has an appreciate barrier with respect to the wild type. In a second step of our investigation, we have studied the effect of the observed mutants and the observed barriers on a simulated dynamic with respect to time when after the absorption of one photon, and for that we used Frank-Condon trajectories. We found that the less fluorescent mutants, the single one, disappears in less than 100 femtoseconds of the excited state, while the most fluorescent mutant, the double one, remains there for more than 200 femtoseconds. To understand if the increase of this barrier is due to a static or electrostatic effect, we recompute the energy profile of the excited for the isolated chromophore, for the isolated retinal chromophore, which was taken with the optimized geometry which was inside the protein. Is exactly the same. It concluded that the increase of the barrier is due to a combination of a static and electrostatic effect, being the electrostatic effect higher in the single mutant and the static effects in the double mutation. Well, now I would like to show some of the most highlight conclusions that we obtain in this research. And I would like to highlight that for me, the most important conclusion was that now with automatic rhodopsin protocol, we are not only able to reproduce the experimental data, we are also able to predict new data for new adenosine rhodopsin mutants that can help the experimental group to generate those mutations and also save time, because we know that time is very important for us during the research. And also, I would like to tell you that this protocol is now using in the laboratory in Japan to produce a new trends of mutants that can improve the current fluorescence. Well, now I would like to thank my research group, the Laboratory for Computational Photochemistry and Photobiology, my supervisor, Professor Massimo Libucci, my colleagues, Laura, Leonardo, Luca, and also I would like to thank our external collaborator that helped us with the experimental data and also with the evaluation of result and analysis of result and prepare of the paper. And of course, I would like to thank you for your kind attention.
Thank you and congratulations again, Maria del Carmen. Now is the moment to present the winner of the third Reaxys SCI Early Career Researcher Award. And it's a pleasure for me to introduce him, Nicola Cavallini from Politecnico di Torino, with an essay entitled Fantastic Online Database and Why Use Them. Please go, Nicola, and enjoy presentation.
Thank you, Emilia. Just tell me if you can see what I'm sharing. If it's okay, then I go.
Yes, it's okay.
Okay, it's okay, I go. Well, thank you. Thank you for the nice introduction and thank you for having me here because I'm so happy to receive this award and for the fact that this is one of the reasons why I like to go out and share what I do. So also being recognized in this way makes me feel very, very good. So my presentation is going to be a little bit different from what you have seen up to now, because my field of work is more concerned with the data analysis than the actual synthesis of substances. And my essay is also my essay was also a proposal more than an actual project. So my idea was to try to link the two worlds of online databases and what I do as a chemist doing data analysis. So let's start with the data analysis in chemistry, which is what I am going to talk about most in this presentation. We as scientists, we collect data in our lab using many different approaches. We do measurements, we collect spectra, signals of very various types, and we hope to be able to extract information from this data. And data is different from information because in data you have noise, you have overlapping peaks, you have confusion, but information is something more structured, something that you can interpret and use to take decisions, to get the knowledge and write a paper, and then build on what we already know in the scientific fields. So what is linking these two blocks? It's some sort of data analysis, probably, something that is driven by data. So we look into the data and we try to connect to the information part. So we try to extract the information part. The field, as we call it, is chemometrics, which comes from chemistry, and metrics, which is measurement. What we do is mathematical models, and we stick to mathematical models that can be interpreted, because the link to reality, so the chemistry, the system, the physics of the system is very important to us. So chemometrics, you can think about it like the intersection between these three other fields: analytical chemistry, which is the reality of the lab, where we collect the measurements and the spectra; statistics, which is represents the instruments, the mathematical instruments we use to analyze those data; and informatics, because we need computers and computational power to build our models and analyze our data. So in the proposal, the idea that I developed for the essay of the REACSIS Award takes the start from NMR spectra. So the analytical chemistry part is represented by measurements with nuclear magnetic resonance spectroscopy. And the informatics part is partially covered by the information provided by REACSIS. So for the experimental data that I used in this experiment, this hypothetical researcher going to the lab trying to solve a problem. The problem is we have 231 mixtures of unknown alcohols. We know that in these many samples, there are only 3 alcohols, but we do not know the identity of the alcohols. So we go to the lab, we go to the spectrometer and collect a lot of spectra. These are the raw spectra, the data, the most complete data set that we have. What we want to do is trying to extract information. And as we can see, and as we know as chemists, information is where peaks are. So in these magnified parts of the spectrum, we have already some information about how overlapped the different signals are, but we cannot really determine if there are only triplets, doublets, or the signals from NMR, those that you can directly interpret when looking at one spectrum. So here comes the chemometrics part, so the data analysis that provides us the tools to extract the information. What if we use something that is able to resolve the curves? And this model that I'm showing to you just briefly depicts the way that MCR, multivariate curve resolution, operates on the data. We have X, which is representative for our spectra, and the 2 matrices that I want you to focus on is the C and S matrix. The information contained into the spectra is divided into relative concentrations on one side and pure profiles on the other side. What we want to achieve is to have pure profiles which are corresponding to the unknown alcohols. So to keep it short, we run the data and through this algorithm to obtain raw data. Now, using the color coding, it's much more easy to understand what was hidden under the initial confusion of overlap spectra. As you can see, just focusing on the left-hand side, we have resolved components that turn out to be just simple signals, 3 triplets, 1 red, 1 yellow, and 1 blue. So the 3 components are related to a single and individual alcohol. And in my thought experiment, we have this researcher that focuses on component number 2 because using his prior knowledge and his experience as a spectroscopist may already know that probably that component is related to propanol. So we go to REACSIS and the most simple query you can ask without any other word. The hypothesis you have and NMR, the technique you have been using so far. So the first match is, of course, propanol, but then the heat data gives you 89 results about NMR spectroscopy. The nice thing is that you already have here the type of NMR information that you can use to confirm and do the peak assignment, which means you also have the references. So by skipping through these 89 results, you may be able to find a spectrum or just the assignments of the signals that you already have resolved using MCR. So this is actually the conclusion of my talk today. And to recap, what I wanted to show is that data analysis starts before actually analyzing the data, because you need to know how to collect your spectra. But also, once you have the results, you need something to validate what you have found. And the take-home message here is that having online databases like Reaxys, which are very complete and very rich in different types of information, should be used even more together with proper data analysis instruments. So thank you all again, and thank you all for the prize to the organizing committee.
Okay, let me thank Nicola for his presentation, and congratulations again. Now it's my pleasure to introduce the winner of the most popular video award of Premio Levi. The winner is Rosaria Bruno from the University of Calabria with a paper entitled Multivariate Metal Organics Frameworks for the Simultaneous Capture of Organic and Inorganic Contaminants from Water, published on Journal of American Chemical Society. Please, Rosaria, the screen is yours.
Good morning. Can you see me? I think yes. Can you see me? Yes. Okay, I can start. Good morning. I am Rosaria Bruno, and I applied to this award as PhD student. Today, I am postdoctoral researcher at the University of Calabria in the research group of Professor Donatella Armentano. It was a very pleasure to participate to this award that allowed me to communicate and divulgate this high-level result with you and with the world community. In particular, the production of a easy video to communicate even on social media. This result allowed me to encourage and drive up the motivation of, for example, young students that want to pursue a research career. So the article mentioned for this ceremony today talks about a multivariate metal organic frameworks. Is a three-dimensional structure of a crystalline material with high porosity, and this material is focused. You can see the presentation in the full mode. I'm asking, yeah, now it's in full mode. Okay. This material is focused in a wider research topic regarding the fight against environmental concerns, for example, gas treatment, and in this case, water remediation. Family of metal organic frameworks based on calcium and copper metal nodes and an organic linker derived from amino acid give rise to a family of biocompatible isoreticular metal organic frameworks that differentiate each other, respect the shape of the pores, functionalization, and surfaces, internal surfaces. The previous result in the capture of heavy metals, for example, mercury chloride, conducted on two different experiments using two different metal organic frameworks based on the methionine amino acid down, and the upper image we can see the other material based on the cysteine methylated, and even for organic molecule dangerous for yield, a series of organic dyes in an experiment of removal showed the high performances of this kind of material that can perform an internment of this species, and the crystallinity of this material allowed the characterization by single crystal X-ray diffraction. And so the insight showed the real interaction, the direct bonding by H bond, for example, between guest molecule and host matrix, sometimes mediated by solvent molecule, in this case water. So the characterization allowed to visualize the interaction and the direction of propagation of the supramolecular chains interned in the channels of this material. Starting from the previous result, the design of a new advanced material, a multivariate metal organic framework in which different functionalities take part in the same building construction. So, starting from the structural characterization, we see interesting results. The diffraction data were taken at the diamond light source, and we can resolve using this data the positional and statistical disorder of the side chains of the amino acid that decorate the MOF pores. The experiment was conducted on some organic dyes and a series of high-toxic metals, and different experiment was performed on single dye and on the mixture of the dyes. In the case of the mixture, the removal was very high, and another conclusive experiment in the removal of the organic dyes and the metals together gave rise the best result. And this conclusive experiment was conducted in a mineral water to miming the real condition of natural resources contaminated. The data of diffraction of the adsorbed mercury chloride and methylene blue inside this single crystal showed initially that after the adsorption of mercury chloride, enough space was available for the capture of the organic dyes. And from this insight, we can see the specific interaction between the nitrogen atom of the methylene blue and the serine residue, and the interaction between mercury chloride and the sulfur atoms belonging to the methylene blue and the methionine arms facing inside the pores. The presence of the mercury chloride confers two different conformations in the distribution of the methylene blue. The high disordered molecule didn't allow the resolution of the amine residues. And just to conclude, we can say that this high robust material was tested and reused for more cycles. It is resistant at room temperature. It's produced by a very easy synthesis at room temperature and then water solvent, so it is easy to scale up. And the perspective is to use directly this material in a filter in the wastewater treatment plants. Thank you for the attention. Thank you for the mention. And I really acknowledge my professor, Donatella Armentano. Yes, and the co-authors of the article with which I participated to this award. Really thank you all. And congratulations for all the winners of today.
Thank you so much, Rosaria, for this very nice presentation. And Now that it's actually the time to thank everyone for being here, congratulate everyone for the prize that they were awarded, and I will just thank also all the board of SCI Giovani, which is present here either as chair or as attendees, and they contribute quite a lot in the evaluation stage and also to make this event happen. And also thank you so much to our sponsor and partner, Elsevier, which is represented here by a few of our members responsible for the Italian accounts. So thank you so much. Also, Thea wants to say hi and thank you to all of you, as you can see here. And let's see each other soon. So hopefully at another event. Thank you again, and bye everyone.